Uh, okay, we'll wait for 1, 2 more minutes before we start. Okay, I hope you can see my screen. Can someone confirm if you can see my screen or not?
Can see it.
Okay,
Yeah, we can see this thing.
thanks. Okay, so hello everyone. Welcome to this kind of crash course on genomic data analysis. And before I start, let me give an introduction about myself. So, I am a second year PhD student at ISC Bangalore. I work, my primary guide is Professor Blatati Kohli from the Center for Brain Research. And my primary work revolves around NGS data analysis, including sequencing data, GWAS data, and like our department specializes in uh, neurodegenerative disorders. So my focus is on mostly on neurodegenerative disorders as well, but I have worked with NGS data all along. So yeah, before I start, so I saw some of the responses of the Google form yesterday. So I kind of have an idea about what you guys actually want out from this course. But I actually saw someone wrote down ki, like they want a job after this. So uh, yeah, so you can like NGS data analysis is is a big thing you cannot learn in 4 hours. So you need to even after this course, you need to kind of go back and kind of try hands-on with real data and stuff. So I'll suggest if you after these classes, if your interest has increased in these topics, please feel free to kind of uh, try all these hands-on because unless and until you do hands-on stuff with this, it's very uh, hard to learn. So for this course, I'll be primarily I've divided it in 2 days. So in the first day today, I'll talk about sequencing technologies and different type of algorithms for data pre-processing and sequence alignment. And for the next class, we'll talk a little bit about how to call variants on genomic data. And after that, we'll do some hands-on analysis of genomic data. So for that part, I'll be working on my system. So my system is a Mac, which is Linux, like where Linux commands work. So most of you will be having a Windows laptop. So if you kind of want to work with Linux in your Windows laptops, I'll suggest you download WSL or yeah, if you have access to a Linux machine, is pretty good. And yeah, so that's it. So I think it's 5:35 already, so I think we can start. Okay, okay. This is not going. Just a second. Yeah. Okay. The most basic sequencing technology that came first was the Sanger sequencing technology. So this was way back in 1977, and Fred Sanger, the person behind the technology, sorry, um, yeah. So yeah, Fred Sanger, the person behind the technology, was actually awarded Nobel Prize for this in 1977. So the technology is pretty simple. So this whole process is the like kind of the overview of this process is called sequencing by synthesis. So what happens is if you have a single-stranded piece of DNA and you add DNA polymerase to it and you add a special type of nucleotides. Like normally we add normally the nucleotides there are DNTPs, your deoxy nucleotide triphosphatase. But what Sanger did was he added DDNTPs, which were dideoxynucleotide. So in deoxynucleotide phosphatases, you'll have one deoxy group, and in case of di, it's pretty evident you'll have two. So the important thing of these DDNTPs, important feature or characteristic, excuse me, characteristic of this DDNTPs are, like, once this DDNTP binds to to a strand, basically if a DNA polymerase is kind of synthesizing the strand, if instead of a DNTP, a DDNTP comes and binds there, the synthesis will be halted then and there. So after a DDNTP has bound to a particular strand, no further synthesis can occur. So what it does is it basically halts your sequencing for a second, like for a second, and you can have an idea about where that particular sequencing stopped. So the last nucleotide will be a DDNTP, and after that, your sequencing will stop. So Sanger overcame this problem of, like, if you just add DNTPs, what will happen is it will add your nucleotides one after another, and you cannot capture what nucleotide is getting added in real time, because that is what you want if you want to know the sequence of the DNA. Now, the workaround behind that was to add these DDNTPs, so this can halt that synthesis, and you can do gel electrophoresis or other kind of techniques to understand how much has at what position the synthesis has stopped, and what is the base that nucleotide that has been added to the position that was just uh, before the position that it has stopped. So the idea is pretty simple. This is called sequencing by synthesis. Sorry, I just need to admit. Uh, yeah. Yeah, so the idea is pretty simple. Instead of adding DNTP, you add a DDNTP into the solution, so that the DNA polymerase, after adding the DDNTP, cannot continue its DNA synthesis, and you can have an idea where that synthesis has stopped, and what position, and what is the last base that has been added. So if you can do this at every base of the nuclear of the whole DNA sequence, you'll get an idea about the whole sequence. But obviously there are drawbacks to it. We'll discuss about it. Uh, so this is a pictorial representation about how this Sanger sequencing works. So you have this is your sequence. This is the sequence that you are trying to get the sequence out of, and onto that you add solutions of normal ATGCs, the bases in DNA, and out of them you add a very small fraction of this DDNTPs, or dideoxynucleotide triphosphatase, which are the special molecules which will halt your DNA synthesis. And then you have the DNA polymerase enzyme. So as we know, DNA polymerase cannot start the replication of the DNA by itself. So you need to add a primer. So a primer is added. And after that, what happens is the DNA polymerase starts synthesizing this strand. So at, say for example, if it comes here to this A position, it will go and pick up a T and add it. So this T might be a DDNTP, right? So if a DDNTP T has been added there, the synthesis will stop. And again, if there are other A positions in this whole sequence, so at the moment a DDNTPT has been added, this synthesis will stop. So for these two cases, these are actually like the pretty rare cases, because the maximum amount of molecules that you have added are normal DNTPs. So at this position, so these two synthesis has been stopped, so that you can, and now you can understand, like, if at this position the synthesis has been stopped, you can have an idea that at this position, since you had added DDNTP thymines, at this position there will be a complementary base to thymine, which is A. So wherever the synthesis stops, you can have an idea what is the complementary base for that particular DNA strand. So you can do it. So at one go, you do it with one type of base. In this case, it was sorry. Uh, in in this case, it was thymine, but you can do this for all types of bases. So A, G, and C. in one iteration you do it for one particular base, so you know at what positions the synthesis stops and what position is that ddntp added and what is the complementary base for that particular position. So this is a pretty like simple idea to go with that. Now after your synthesis has been stopped, you get like the half double stranded DNA like this. So you have one full strand and one half strand like this. It'll stop at the position that ddNTP has been incorporated. Now, how you can differentiate between, say, for example, this double strand and this double strand. So you run it through a gel electrophoresis. What is a gel electrophoresis? It's basically uh, a mechanism by which you can separate molecules based on their molecular weight. So if a molecule is having higher molecular weight, it will travel less in the gel electrophoresis column. If it is having a lower molecular weight, it will travel higher up in the gel electrophoresis column. So your the kind of output that you have in gel after you run this through the gel electrophoresis columns is something like this. So for each column you have a particular base, and at what position this particular base has been added, you can have an idea about the molecular weights distributed across all these particular strands. Now, since this is a pretty simple, the idea is pretty simple, but there are some issues with this. So this becomes challenging to kind of resolve two particular strands which are having very similar molecular weights when their individual molecular weights are very high. Say, for example, it is very easy to differentiate between a DNA strand of having like say three base pairs and two base pairs, but it is not that easy if the DNA strands are having thousand and one base pairs. So one of the major limitation of this Sanger sequencing is this works roughly below 700 base pairs. It won't work beyond that because as the length of your DNA increases, the L increases, the length of the sequence. The distinguishing factor between the mass of a sequence of length L and the mass of a sequence of length L plus 1 becomes increasingly harder. So this was pretty so this was actually the major issue with Sanger sequencing, that you cannot sequence long stretches of DNA together. So that is one of the major issues with Sanger sequencing. Now, one more other type of sequencing that was there after Sanger sequencing, before we come to next generation sequencing, was DNA microarrays. This idea is also pretty simple and elegant. So you have a DNA microchip like this. You have a chip. In this chip, you have uh, this particular oligos, or basically your probes. So basically, these probes are single-stranded DNA that are complementary to the DNA that you are trying to find. So basically, this works very well in RNA sequencing technologies because, say for example, you want to see how much a particular gene is getting expressed in a cell or in a tissue. So what you do is you extract the DNA from the cell, and then you uh, try to break it down into small pieces, and then you put the whole DNA on this microarray chip. But this microarray chip will contain only complementary sequences of DNA which are complementary to the gene you are trying to find, or the gene you are trying to have an idea about its expression. So only those complementary strands will be there. So if your gene is getting expressed, your DNA or your RNA, whatever you're trying to look at, will come and hybridize with this particular probes that are present inside in the microarray. So this is a very like a very elegant technique to do that, because it is, firstly, it is very cost effective, but and secondly, it is easier to do. So, but there is a very big but along with it. He, you cannot, already have to know what you are looking for. Then only you can try a microarray, because these probes that you design on the DNA chip, on the microarray chip, needs to be pre-designed by considering the fact what you are trying to achieve. So, if you are trying to see the expression of a particular gene, your probes need to be kind of complementary to your the gene of your interest. So this is also a pretty widely used method. If you will see a lot of RNA sequencing, not RNA sequencing, I mean gene expression data being generated by microarray technologies. And after microarray technologies, we moved on to NGS. But before NGS, this is a very widely used method that was prevalent across sequencing technologies. Right. Now, as I was saying, the principle of DNA microarray technique is pretty simple. It lies on the hybridization principle between nucleic acid strands. So basically, you have a complementary strand in your microarray, and the other complementary strand from the cell that you have isolated will come and go and bind to it. Now you can have fluorescently labeled target sequences that bind to a probe after it has bound to a probe signal will give you a particular type of fluorescent signal. Those kind of things can be incorporated in your DNA microarray chip. But yeah, this is an overview of the idea about how DNA microarray works. So you need to have an idea about what you are looking for and create complementary strands which will act as probes on the DNA microarray chip. And after that, you can see how much of the DNA in your cell or DNA or RNA in your cell is coming and kind of binding with the probes. So you can have an idea about the gene expression or about the particular stretches of DNA that is present in your sample. Now, if we move on from, okay, so there are a couple of advantages and disadvantages of microarray. As the advantages is basically it can generate uh, single experiment can generate a lot of results very easily, and it is fast and it is easy to obtain results. But the major drawback is it is the microarray. If you're creating a custom microarray chip, it is quite expensive. But there are like standard microarray chips that are available from different companies. But if you're creating your own, it's quite a time intensive and very expensive process. And these chips do not have a very high shelf life. So once you use it, you need to kind of like the next time you use it, you might need a new chip. So in that terms, it becomes a little bit expensive. But if you compare this with NGS technologies, it's quite cheaper, right? Okay. Now we see Sanger sequencing, then we see microarray technologies, but both of them are actually having a lot of drawbacks. So that's why we move on to next generation sequencing. So this is NGS is a very commonly used catch-all term for all types of sequencing. It can be DNA sequencing, it can be RNA sequencing, it can be some sort of protein sequencing you are doing. So this NGS is a catch-all term for all types of sequencing. We in this course will be focusing mostly on genomic sequencing. I have not included RNA sequencing as a part of these two lectures because it'll kind of extend the lecture a lot more and it'll kind of confuse you as well because it is not possible to do that within 2 days, 2 hours of lecture each. So I've not kept the RNA sequencing part in this whole in this whole course. But yeah, we'll be mostly focusing on genomic genome sequencing or DNA sequencing. Okay. Okay. So the first type of NGS, I just can't able to move this. Yeah. The first type of NGS we'll be discussing is Illumina sequencing. This is one of the most commonly used sequencing method for sequencing DNA and RNA. Now, this principle, the principle that we saw on how Sanger sequencing works, the principle of Illumina sequencing is pretty similar. So this Illumina has so let me go through the whole process. So firstly, the major advantage of Illumina is that it achieves parallelization by running several synthesis experiments. By synthesis, here we mean sequencing by synthesis. It's basically a sequencing experiment. So Illumina can sequence multiple stretches of DNA all at one time. So it achieves a massive parallelization in that front. Here also DDNTPs are used, but with a twist with a fluorescent tag. So the concept is similar that every time a DDNTP is added to a DNA strand, the synthesis will stop, and uh, and along with the synthesis getting stopped, since you have added a fluorescent tag to it, it'll emit a fluorescent light. So each of these DDNTPs will be incorporated with a fluorescent tag, which will emit different wavelengths according to the base that is getting added. So when A is getting added, you'll have a different wavelength of light. When T, G, C, whatever is getting added, you'll get a different wavelength of light. So basically, these frequency signals act as signatures for this particular basis. So if it is a particular type of signal coming from the sequencing machine, you know, or the base caller knows technically that that particular signal is because A or T or G has been added, right? So these are very specific signatures for particular bases. Depending on that, you add the fluorescence tag. Uh, all new strands are of the same length, and at the end of each synthesis cycle, after the end of the synthesis cycle, a picture is taken. A picture of what? A picture of that fluorescent wavelength of light getting emitted after a particular base has been added. So again, this picture is what actually the sequencing machine takes, and after taking up this picture, there is a base calling software either within the sequencing machine or some like in a computer there. The base caller looks at this particular picture and predicts what base has been added at that particular position of the DNA. So what happens in Sanger sequencing is, like, once you add the DD and TP, your synthesis completely stops. But here, what they do is reversible termination. So once your DD and TP has been added, the DNA polymerase cannot continue synthesizing the strand. Now, what will happen is you add some sort of an enzyme that can convert this DDNTPs to a DNTP, and after it has been converted to a DNTP, it can again, the DNA polymerase can again start synthesizing the strand. So here, what the technique is called reversible termination. So the idea is you do not stop at after adding a DDNTP. You After you have gotten the signal that a DDNTP has been added and this particular base has been added at that position, you immediately convert the DDNTP to a DNTP so that the next base can be added, and in the next cycle you can get an idea about what is the next base that has been added. So this process is called reversible termination. And for high-seq is a type of an Illumina sequencer machine. Each of the cycle takes about 6 minutes. So it's actually pretty time consuming. Okay. Yeah. So this is how it actually looks like. So for each, say this is your probe, a particular base has been added to this probe, so it will emit a particular light. Again, for each of the probe, whatever base has been added to that will get a light of that sort. So, for example, green has been added up to it. So let's assume green is a signature of A, and yellow is a signature of T. So in this particular cycle, a green and a yellow has been added. So after the sequencing machine takes a picture, the base caller can identify that the green has been added at the first position. That means this position, at the first position, A has been added, and what is the complementary to that? So that is your original DNA sequence. Similarly, at the first position, yellow has been added. Yellow is a T, and if T has been added, what is the complementary to T that is present in your sequence, which is going to be A. So like this, for each cycle, a particular DDNTP is added, which emits a fluorescent tag, and that picture is processed by this particular sequencing machines, and the base callers predict what base has been added to that particular cycle. So I'll play this video actually. I'll play this video. So this is actually the official Illumina sequencing workflow video shared by Illumina only in the YouTube channel. You can go and take a look at this. I'll keep this in uh, the chat box as well. So if you want to take a look, please. Oh, sorry. Um, just give me a second. yeah, you can take a look. I'll play this video here because it's a 5-minute video, but it actually explains the Illumina sequencing pretty clearly. So I'll just play it here.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a
So this is actually the flow cell inside which those probes are kept, and inside this only the DNA polymerase is also there, and the ddNTPs are also there, which keeps getting added. So this is actually the place where all the synthesis occurs.
process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with alon, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface.
So the adapters that you add in during the sample preparation step is actually these are the adapters that are going to find the complementary oligos that are present inside the flow cell and go and bind to that. Okay, so the adapters and the extra bit of DNA that you are adding on both sides of of your original strand is because so that it can go and attach to that particular oligo, complementary oligo that is present inside the inside the flow cell.
This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over.
So this is the process of bridge amplification. So the 2 ends of the DNA that you have added primers. So one will be complementary to the one that is already there. The other will be complementary to the other type of oligo that is there, so that it can go and bind to the other oligo, forming a bridge between these 2 oligos. So after formation of this bridge, DNA polymerase will synthesize the new strand, and this is how your DNA is amplified inside the cell.
Over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming.
Now this is the portion where actually the sequencing occurs. You have the sequences, you have added the primers, you have amplified the DNA across. Now you want to synthesize the DNA, or like kind of sequence the DNA by synthesis. So yeah.
Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain.
Exactly. So if this T has been added, say for example, a red signal is emitted, you know a T has been added, and you know what is going to be the complementary. So at that point of your sequence, you know there is an A. Like this, the base caller also knows in what position a particular sequence nucleotide has been added, and what color has been emitted, and how you can trace it back to the original sequence.
Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously.
Yes. So one important thing to remember is the cluster amplification that you are doing, or the bridge amplification that you are doing. In part of it is actually due to the fact that because if only one particular base is being added at one particular position, the amount of signal it will generate is very low. So it's very hard for the sequencer to capture it again. So it's better to have multiple copies of the same DNA, and at every point, if a particular base has been added or a nucleotide has been added, you can get the signal from across all those particular amplified DNA stretches. So your signal strength will be much stronger. So that is also one of the reasons why you do bridge amplification.
Hundreds of millions of clusters are sequenced in a massively parallel process. This image
Yeah, this is the image the sequencing machine captures. So at every position, you know what base is there, at in what flow cell, you know from what flow cell that signal is coming, and the base caller will know if that particular signal has come, what is the complementary strand for the DNA.
represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away.
Now you see the same DNA strand is being synthesized in two different manners. One from starting from 5' to 3', and the other in the opposite direction when you have done that bridge amplifications thing. So for a Illumina short read sequencing data, what you'll see is two reads. There will be a forward read, there will be a backward read. So there will be R1 and there will be R2. So for Illumina, generally two FASTQ files will be generated, or two basically sequencing files will be generated, one coming from read one coming from read two. So this is known as actually paired end sequencing. This is a term that will use paired end, and why we require paired reads and how we use the concept of paired reads in determining variants. We'll come to that in the next class. But yeah, this is how your two types of reads are generated. You have the same strand. Once it is getting synthesized in this manner, then you are washing it off. You are putting it onto the other oligo and kind of reversing the strand and then reading it again. So the same strand is being read from two different ways. So you'll get a read one generated and also a read two generated from the same strand. Right?
Leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments.
Okay, I'm not going into the data analysis part because we'll cover this according to our own. But yeah, I just wanted to show you actually how this Illumina sequencing works. And because Illumina is like the kind of the most widely used sequencing platform, so I thought, okay, I'll go into a little bit more detail for Illumina. But sorry, yeah, okay. Now let's move on to other types of technologies, but uh, the concepts for Illumina for paired end sequencing will come in handy when we do variant calling or structural variant, particularly structural variant calling, right? So please keep that in mind, how the two reads are generated from the same strand, right? Okay, moving on. So as obviously there will be limitations to these type of sequencing also. Firstly, it is limited by the size of the fragments that can be sequenced. So there are two types of technologies for NGS sequencing. One is the short read sequencing, one is the long read. So Illumina, this type of sequencing is called short read sequencing, where you can sequence only short stretches of a DNA at one particular point of time. So this So you're sequencing with read one and read two, two reads from the same sequence are added up to around 300 base pairs. So that is the average read length for Illumina sequences. But when we see about long read sequences like Oxford Nanopore or PacBio, this read length increases by a lot. This goes up to over 1 KB, sometimes 10, 15, 20 KBs like that. So that is a much bigger length of sequence that can be sequenced at one particular time rather than Illumina, which can sequence only a short stretches of DNA at a particular time. So that is the first disadvantage of Illumina. The second is that it can take a long time to generate. As I was saying, the time required is very high, and also you cannot do real-time data analysis because you do not have access to real-time data after the sequencing step has been completed. After the base caller, then only you can use the base caller to identify the sequences and then do your analysis, right? So you have to wait till the whole experiment gets over and takes a little bit time. So that is kind of a disadvantage for Illumina. Okay, so the major disadvantage for Illumina, as I was saying, like is the sequence length. It's only 300 base pairs. But if you see about long read sequencing technologies like SMRT, so the full form of SMRT is single molecule real-time sequencing, right? So this is also the principle of sequencing is also the same. It is done using sequencing by synthesis, but there are some special terms here. So first is this DNA synthesis, as Illumina Illumina we saw it is done in flow cells inside the chip, but in this case it is done inside some DNA wells which are known as zero mode waveguides. I'll not go into details, but this type of wells are used because it can concentrate a particular signal that is coming from inside the well to a very good level. So you can read about ZMWs a little bit more if you are interested, but the idea is same, that you have wells inside the inside the in sorry yeah, you have well-like structures inside the flow cell, and inside the well-like structures your DNA strand will go inside that, and after that the DNA polymerase will synthesize the DNA as before, as in Sanger or as in Illumina, but in this case also a particular tag has been used, which is the fluorescent tag. Now these wells are constructed in a way that only fluorescence occurring from the bottom of the well is detected, and the fluorescence label detached from the nucleotide at its incorporation into the DNA strand, leaving an unmodified DNA strand. And this SMRT technologies allow for sequencing for close to 1,000 nucleotides together. So it's 1 kb. So it is actually long read sequencing where you can sequence a stretch of 1 kb length at, at one stretch, right? But the principle is pretty similar. But instead of those flow cells, these SMRT use zero ZMWs, which give you this advantage of being able to sequence a stretch of 1,000 or more nucleotides together. So coming to the principle of this SMRT sequencing, so what happens is this whole thing is occurring inside the well. Remember that. So this is your original DNA strand. This is your DNA polymerase, which is kind of binding the nucleotides complementary to your strand. Now these again are tagged with fluorescent dyes, which can be pictured by the sequencer. Now once the DNA polymerase adds that particular nucleotide, a particular fluorescent signal will be emitted, and the signal from each complementary strand is read as it is added to the sequence fragment. And so this is your sequence fragment, and this is your original DNA strand. So the principle behind it is pretty similar, but because of this ZMWs and the technology behind it, you can sequence a stretch of 1,000 or more nucleotides at a time using SMRT. Right. So moving on, another type of long read technology, uh, but like this is kind of in the middle between long read and short read, is basically ion tolerant. So this from all the technologies we have studied, so Sanger uses some sort of fluorescent tagging or some sort of DDNTPs to stop the synthesis. Illumina uses fluorescent tagging. SMRT also uses fluorescent tagging. In this case, in ion torrent, the signal that is generated is not fluorescent light, but it is actually the change in pH of the solution after a particular base has been added. So after, say, addition of A, T, or G, or C, the amount of pH change in the solution will be very kind of it will be a signature of the nucleotide that has been added. So, for example, A has been added, the pH change will be of a certain level. If T has been added, the pH change will be of a certain level, and so on. So, the principle is when a complementary nucleotide is incorporated to a strand of a DNA, a phosphodiester bond is formed, releasing an H plus ion into the solution, which changes the pH of the medium. The ion sensor will convert this chemical information into digital electronic information, just as the base callers they do as well. Check what light has been emitted, and then you can convert it into the digital information about what base has been added. Okay, so yeah, this is the whole overview. So you can have you'll have your own stretch of DNA. Now on top of this, the DNA polymerase will add one after the other the nucleotides, and after addition of each nucleotide, there will be a change in pH, which will be signatory of the base that has been added. And after this, you can have this is what the output will be. So for each change in pH, you can attribute to that to a particular base that has been added, right? So we saw like if you notice, all of them are actually using sequencing by synthesis method. So you are sequencing the DNA by synthesizing the complementary strand of the DNA. The kind of signal you're using to identify what base has been added, that is different across the technologies, but the whole principle is kind of similar, that you are sequencing the DNA, the complementary strand of the DNA, to know what is the actual sequence of your DNA stretch. Okay, now we come to Oxford Nanopore technology, which is kind of the state of the art now. PacBio is also there. PacBio with its hi-fi reads, high fidelity reads. You can go look that up as well. But yeah, ONTA and PacBio are the kind of the major long read sequencers available, like used majorly. So actually, ONTA is kind of a little bit different to how it works compared to Illumina or Ion Torrent or SMRT, PacBio, etc. So in this case, the principle is that the DNA can be sequenced by threading it through a microscopic pore in a membrane, and the bases are identified in a way that affect the ions flowing through the pore from one side of the membrane to another. So this time you are not sequencing any kind of DNA, but what you are doing is you are passing a DNA through a protein and through a pore in the membrane, and also you're passing current along with it. So change in current for each base will be different. So if a particular, say for example, in the first stretch, this nucleotide comes along and the change of current is say for 0.2 ampere. I am just like telling you random units here, just for example. So do not take this as like kind of original units or something. So for example, your A, when there is a A inside inside the protein, the ion current flow change will be around say 0.2 ampere. If that is a C, maybe it's a zero point 0.5. If that is a T, if maybe it's a zero point 0.1. So that also there is a particular signatory amount of change in current for each base that is present inside that particular protein. So this is how your protein looks like. So if you guys have read about respiration, this kind of looks like a F0 F1 ATP synthesis, right? So yeah, you have basically two proteins on top of one another. So one protein will unzip the DNA helix because it is a double strand, and only one strand will go inside the protein through the membrane, and the second protein will create a pore inside the membrane and will hold the adapter together. Will hold the DNA together with the help of an adapter. So the adapter molecule is attached here, so that it keeps the DNA inside the membrane, inside the protein, and through to the membrane. So ONT is a very widely used technology, and it can generate very long reads of sequences together. So like if you are doing some sort of structural variant calling, or like where you require or you are detecting duplications inside the genome, This long read sequencing technologies help in the form that like where there are duplications or there are large stretches of the genome where there are duplicated sequence. Short reads, if you are having a read of say 300 base pairs, and your stretch of the DNA that has repeats is around, say for example, say 10,000 base pairs, then the 300 base pair, it's very hard to understand where that 300 base pair of read came from inside the whole 10,000 base pair region. So when there are repeats and there are duplications inside the genome, long read sequencing help us to kind of navigate through these duplications and align the reads properly to the reference genome. So but then again, long read is much more expensive than Illumina or other short read technologies. But yeah, it is actually from a biological perspective, it is more of use than short reads of data like Illumina. Right? So these were all the sequencing type of technologies that are currently in use. There are other technologies as well. I also did not discuss hi-fi reads. You can maybe probably look that up. It's also interesting. But yeah, these were the different types of technologies starting from Sanger, then to Illumina, then SMRT, then ion torrent, then ONT. So these are the different type of technologies that has been used across the years to sequence DNA. Now, after you have done the sequencing, after you have identified what base is at what position according to your reads, so the reads that you have, the stretches of DNA that you are synthesizing, I hope you have understood by now that they are not the whole DNA sequence. They are stretches of the DNA sequence. You break down the DNA sequence into small stretches and you only synthesize the stretches. Now, the stretch size may be smaller or bigger depending on your sequencing platform, but it's always a stretch of the DNA. The whole DNA cannot be synthesized at one go. We do not have any kind of technology for that. Even ONT cannot do that. So, uh, what happens next is, so you started off with a sample with the whole DNA, you broke that DNA off into small pieces, and you sequence these small pieces of DNA. Now, the next step is to know from where these particular sequences of DNA generated in your original genome, because you only have stretches of the DNA, but you do not have any idea where those stretches came from. Now, to do that, we'll follow a series of steps and ultimately do alignment. But yeah, bear with me. Now we are moving on to the next step. After your sequence, how do you identify where that particular read came from? Okay, before we move on to that, a couple of technical terms. What one is sequencing depth, one is sequencing coverage. So what is sequencing depth? So as I was saying, use sequence stretches of the DNA, not the whole DNA. Now, these stretches of the DNA, say for example, from base pair 1 to 1000, you're sequencing that, but you're not only sequencing only one read of that 1 to 1000, you will be sequencing multiple reads of the same stretch of DNA because you'll amplify your reads and you'll sequence each of those reads together. So the sequencing depth is the concept, or it is also known as read depth or depth of coverage. You'll find a lot of terms out there, but what it means is actually the number of times a particular nucleotide or a particular base in the DNA is read during the sequencing process. So for the stretch of say from 1 to 1000 base pairs, you have say 50 reads. So all the bases inside that stretch, 1 to 1000, all the bases have been sequenced 50 times. So your sequencing depth will be 50 there. So the higher the sequencing depth, the more the times you sequence a particular region of the genome, it's actually better because the less chances of error and noise that you'll get. And also, like the standard sequencing depth used for human genome experiments is around 30x. 30 to 40x is the average sequencing depth you'll find, but this depth will differ from platform to platform. Some platforms require higher depth, some platforms require lower depth, but around the 30x mark is what you'll find your sequencing depth at. Now that goes sequencing depth. Now what is sequencing coverage? Coverage is basically the proportion of the genome that has been sequenced at least once. So, as I was saying, 1 to 1000, maybe you have sequenced it 50 times. But it is possible that 1000 to 2000, maybe you have sequenced it only one time. Maybe at another stretch of the DNA, say from 1 million to 2 million, you have not sequenced at all. There is no coverage there. So, what sequencing coverage means is the proportion of the genome that has been sequenced at least once. So at least once has all your DNA, all the sequence, all the stretch of DNA has been sequenced or not. So for example, 95% coverage means 95% of the intended region has been sequenced at least once. So this sequencing, this depth of sequencing will vary from region of the genome to region of the genome. But the idea is to kind of uh, sequence the whole of your genome at least once. So that is what by coverage we mean. So coverage and depth are very like interconnected in nature. So by increasing the sequencing depth, you can also increase the coverage because more reads mean a higher likelihood of covering regions. So it's basic probability. However, due to biases in the library preparation, certain regions may still be underrepresented or missed entirely. So even if you increase your sequencing depth, you cannot increase it to like infinity and hope that all the portion of the genome will be covered. It does not work like that. It will increase the coverage to a certain level, but it won't like kind of cover for it, right? So you need to have coverage across good coverage across the genome. Now, these concepts of sequencing depth and sequencing coverage is important mostly when we look at variant calling. So if a particular, so I hope you're familiar with the idea of mutations. So basically that can be like one or multiple changes in the genome simultaneously. So to identify if there has been a change or is it actually the original genome, you need good amount of evidence, and that evidence is provided by this sequencing depth and coverage. So if a particular portion of the genome is an SNV and it is supported by say 50 reads, you will be more confident than an SNV which has been covered by only one read, right? So variant calling is very important. Sequencing depth and coverage is very important between for variant calling, and there also needs to be a balance that is struck between depth and coverage. You do not want like excessive high amount of depth and low coverage, or excessive high amount of coverage, I mean like 100% coverage and very low depth. So both you need to have a balance between that, right? Okay. So yeah, this is what I was talking about. So this is your depth of coverage, a particular region of the genome, how many times it has been sequenced, and this is the breadth of coverage, which is actually just the coverage itself, is how is all the genome or all that intended part of the genome has been sequenced or not. Right. So this is a pictorial representation of actually what it means. Okay. Now you have done your sequencing. You have got the data. Okay, at what position, what base is there. Now, when the base caller software is identifying the pictures from the sequences and predicting a particular base at that particular position, that is obviously prone to error, right? So what we have is an associated quality score associated with every base that has been sequenced. What is the quality score given us? So it's a pretty simple minus 10 log e, where e is your error of probability. So this q value, which is the qual value or the Fred score, quality score as known as, is given by this form like minus 10 of log e, where e is your error of probability, right? And what does like actually it means? So if your q is 1, what does it actually mean? So if your Fred quality score is 10, if you can just calculate E from here. So if it is 10, it's 1 by 10, which is 10% error. If it is 20, it is 1%. If it is 30, it's 0.1%, and it goes on. So anything above 50 and 60, it's like you can assume that it's like mostly error free, right? But yeah, this is how the quality scores work. So for each base called by the base caller, there will be an added quality score assigned to that particular base. And seeing that quality score, you can have an idea what is the probability that that particular base has been called wrongly. Because the sequencing machine can get things wrong. So that is part of the job. So you need to have an idea about how much wrong it is. Like if it is like a 50-50 thing, you would not take that base into consideration, right? Because it'll have higher chance of accuracy, higher chance of error. Right? Okay. Now uh, we come to different type of data formats, right? So before we kind of go into the things that we can do with the data, let's look at the certain types of data formats. So FASTA is one of the most simplest data formats for genomic or proteomic data. So it's pretty simple. You have a file like this. On the header front, you'll have a line which is basically the identifier of the FASTA file. It starts with this greater than sign, and below that will be the stretch of the like sequence of the, in this case, the DNA that has been sequenced, or it can be proteins as well. So as in amino acid sequences. So amino acid sequences can be also represented in um, one letter form. So it can be amino acid sequences as well. So for in this case, in this example, the identifier is this particular number, and the remaining line of the first line, which is the identifier line, are further comments about the sequence, but these actually are not very much required in the downstream processes. It's just giving you the information about like what is the species or what from where of in the genome this has been sequenced or something like that. Information that you want to give before the actual sequence of the DNA. But one thing you'll notice here is this is a DNA sequence with ATGC, but you'll see here is the maximum of the letters are capital in nature, but in the middle, in some cases, it is small. So what is this doing is basically this concept is called as masking. So what is happening is if if a portion of the DNA sequence is falling inside the repetitive region, so that portion is actually masked to represent that portion is a repetitive region. So instead of having these capital letters, they are written in small letters. So this is called as soft masking. If you completely want to remove that sequence from your file, FASTA file or whatever, you can just replace this particular stretches of DNA with N, right? So there will be ATGC and N will be missing, right? So in cases of gaps or in case you want to do hard masking, this is called soft masking, converting from capital to a small letter. But if you want to do hard masking and if you want to remove certain portions of the genome, you can just use N. Right? Okay. The other type of format is FASTQ, which is actually which is known as the Illumina format. The Illumina data comes in a FASTQ format. So FASTQ format is also kind of pretty similar. You'll have a identifier line followed by a sequence, right? But for each sequence, it is a little bit more complicated in case of FASTQ. So you'll have an identifier line, you have a DNA sequence. Next to that line will be almost always a simple positive line, meaning that is joining these two stretches of the DNA. And after that line, so this line is basically this line you see is basically an ASCII coded quality score version of it, like, right? So it's an ASCII coded Fred scaled base quality score. This gives an estimated measure of certainty about each base call in the sequence. So each of these letters A, F, J, say for example a negative sign or a less than sign, all of these represent a particular number in ASCII scale, right? So these are ASCII coded, but what they're representing are specific numbers which tell you and what is the quality score of this particular base at this particular position. So yeah, this is how it looks like, and it like FASTQ starts with a at the rate compared to a FASTA which starts with a greater than sign. Right? Okay, yeah, this I have explained a little bit more here. So so this number K00364 is actually the unique sequencing machine ID. Then the delimiters are colon. Like after the colon, there's a 64. The 64 represents the run number. Then there is another string given with this, which represents the unique flow cell number. Then there is the lane number. This one is giving you the lane number, and then the tile number and the x coordinate and y coordinate of the tiles. So this is the tile number and this is your x and y coordinate. And also, if the sequence is coming from read 1 or read 2, as I was discussing in Illumina, you'll have paired end data from the same sequence will be sequenced from two opposite strands, which one will be read 1, one will be read 2. So if it is coming from read 1 and read 2, that information is also coded there. Okay. Now we have come to this point. So we have gotten into the sequencing. We have gotten some signals. We have done base calling. We have seen the scores of the quality scores of the bases that has been generated. Now you have gotten the FASTQ file. After the FASTQ file, you have got you need to do some sort of quality control. Like, is the read quality is okay? Is there any contamination, or is there any other kind of complexities? Say, for example, it can happen that RNA contamination has occurred in your sample, so you have some RNA sequences inside in your data as well, and you can have overrepresented sequence. You can have different types of uh, issues with the data as well. So these can be this whole like set of quality check parameters can be done through a software called FastQC. So that is also a very it's actually a pretty old software, and it's still in use till now. So the kind of uh, report it gives. So FastQC, tomorrow we'll see how the FastQC report is generated. It gives you an HTML file which you can open in your browser. So yeah, this is the basic statistic part which it will give. It will give the total sequences present, which is the sequence length, what is the GC percentage. I hope you know that all the species will have a certain level of GC content, which is characteristic of that particular species, of the genome of the particular species. Also, along with that, you will have per base sequence quality. So you can see if your per like in each of the bases present, what is your sequence quality? Is it like this is actually not a good plot for this, because you see in some cases you have very high sequence quality, in some cases you have very low. So this is actually not a very good data to go on about. So these kind of information you can get using FastQC. Other information include your GC content, how it should be. The theoretical distribution is the Poisson distribution, and what is the actual distribution of GC inside your data. Those kind of things you can have an idea about, is your data quality okay or not, right? And also overrepresented sequence, maybe there is some sort of duplication inside the sequence. Also, for example, this sequence has been counted for over 2500 times, but there is also no hit. Now, this might be due to some sort of contamination or some sort of PCR amplification that this read has come into your data, but there actually has you do not know from where it came, but it is overrepresented in your data a lot of times. Okay, now after you have seen this kind of plots, this kind of overview about your read data, now what you can do is perform some sort of QC on this data. So these are a couple of softwares that actually do this kind of QC. One is Picard Tools, one is SAM tools as well. So one of the major things to remove is basically duplicate reads. So same reads that has been read twice, it is technically increasing your sequencing depth, but is actually not helpful because it's the same read that has been sequenced twice. So the idea is to identify this duplicated sequence and remove this from your data so that you can have a cleaner version of the data. So Picard Tools does this using mark duplicates function, and also SAM tools does this using this marked up function, marked up hyphen R, which is the remove function. So after you check your data quality, then you do some in intermediary pre-processing, which is removal of duplicate reads from your data. After that, as you know, when you're sequencing the data, there were like adapters getting attached to the ends of the 3' and the 5'. And if you remember the video in the Illumina video, so what can happen is when you're sequencing, the adapters might still be on your uh, uh, on your strand, and while sequencing, this adapter is also sequenced. So what you get is extra data, but those data is not originating from your sample. That data is coming from extra stretches of adapter that are present while you are sequencing. So it's a good idea always to remove these adapters. So you have, for Illumina, you'll have a specific set of adapters, sequence of adapters that are present. So you can search for those sequence of adapters inside your data, and if you find those adapters, then it's a good idea to remove them. And softwares like Trimomatic and adapter removal, these are quite often used softwares which can trim this excess adapter sequences and kind of cut it off from the data so that you can have a cleaner version of the data. So let's go over through the QC steps once again. So first you do duplicate removal. Now first you see what is actually happening in your data. Is your data good quality or not, or want to do anything with it? After that, you search for duplicates and you remove them. After that, you search for adapters that are contaminating your data. You also try to remove them. Next is step is to finding if there are low quality bases in your data. So after the base caller has given a Fred scale quality score to your to each of the bases that has been called, you know what is the percentage of error a particular base will have. So you can have a threshold. Say for example, you do not want to you do not want to take into account the bases that has been sequenced with a quality of 20. You want to remove everything that is below a quality of 20. So you can take a cutoff and you can remove the subsequent extra data there. Subsequent, not the extra. It's actually the low quality data from your original data, and then what you will be left with high quality data. Yes. Okay. Okay. So yeah, this part brings us to the end to the first step of pre-processing. So we started with Sanger. We then moved on to Illumina, then different types of long read, including ONT and TagBio and different stuff. That was how you read the bases, how you generate the sequences, right? Then there will be a base caller that will identify the signals inside this sequencing platforms and generate the reads. After the read has been generated, you use softwares like FastQC to check if the data that has been generated is good quality or not. If not, you do some preliminary QC like removing low quality bases, removing adapters inside your data, or removing duplicates like this. Okay, so this is the first step of pre-processing, and after all these things you have done, what you want to do is to do sequence alignment. Now you have reads, as I was saying, if you remember, you have reads from your data, but you do not know where these reads originated from. You want to know that actually where actually is this read coming from. That information you don't have. So for that, you need to do sequence alignment. So for sequence alignment, what we do is generally we have a reference sequence that has already been generated. So the first human genome project that ended in 2000, I think, if I remember correctly, in 2000. So that generated a complete reference of the human genome. And it was obviously not complete because it is still getting completed. Initially, I think a year or a year and a half back, the T2T consortium came out with a paper where they resolved the Y chromosome regions and the telomeric and the centromeric regions of the genome, before which we did not have much idea about the Y chromosome sequence. We had a sequence, but since Y chromosome is full of repeats, it's very hard to resolve what is actually the sequence of the Y chromosome. So that has been sequenced very recently, like 1.5 years. It's around like 22 years after the first human genome reference we had. So it's actually pretty difficult to kind of resolve the whole genome, but we are right now in a position that we kind of have resolved the whole genome, but still there are regions of the genome where we are a little bit iffy. We do not know if it is actually, we are not confident on particular regions of the genome, right? Now, uh, we have this reference genome, which is a already sequenced uh, uh, like a whole sequence of the human genome, for example. So human genome will have 10 to the power 9 base pairs, 3 into 10 to the power 9. So it's 3 billion base pairs inside the human genome. And you only have reads of 150 base pairs or 300 base pairs. If you combine read 1 and read 2 from Illumina, that together comes up to 300 base pairs. Now, if you are given a sequence of length 3 billion, and then you are asked to find, okay, where is this 300 base pairs coming from? And it is this 3 billion bases is just a combination of ATGC, right? So there is not much patterns in there. There are patterns, obviously, but not very distinctive patterns in there. So it's very hard to kind of find where this particular read came from. So that is what we call as the alignment problem. So we'll discuss a little bit about the alignment problem. Yeah, okay. So first of all, what is the alignment problem? So as I was saying, you can, from a computational perspective, you can imagine it to be two strings you are given, and you are asked to identify where in the first string where from in the first string has the second string been generated. So for example, you have two protein or DNA sequences. You have a stretch like this, x1, x2 up to xm, and y1, y2 up to yn, right? So for DNA sequences, it will be like a TGC, and for protein sequences, you can have 20 amino acids which are represented by single letters. And the output that you want is the optimal alignment between the two sequences to form a list of column operae. So if you're given X like this and Y like this, you want to find what is the optimal alignment between these two particular sequences, right? Now there can be uh, many problems arising due to this. First of all, there can be mutations inside the genome. Like there can be substitutions, deletions, insertions, inversions. Any type of like variations in the inside the genome might hamper your alignment strategy, because you want to see this. You have a read of, let's say, 300 base pair, and you want to find where exactly is this 300 base pair coming from. Now if it is not matching, if 300 out of 300 bases are not matching inside your from your reference genome, then also there can be a good alignment, keeping in mind that there can be these kind of mutations inside the genome. So it is obvious to the fact that maybe 300 out of 300 bases will not match. Say for example, 280 bases will match, 20 will not match, or something of that sort, right? So what kind of mismatches can happen? Mostly insertions and deletions. So if an insertion, if a deletion is happening, so basically there is no, if a deletion has happened in your genome compared to the reference genome, so there will be no portion of in your reads that can actually go and align to that this particular area of the genome, because this has been deleted from your genome. The original reference genome has it, but your genome does not have it. Again, for example, if it is an insertion, then your genome or your reads will have that sequence, but your reference genome won't. Right. So these kind of mutations can have varied impacts in your in the functionality of the genome, but we are not going to go much into that. Right. Now, if you are considering like aligning a stretch of the DNA to a long stretch of the genome, then you need to account for these particular mismatches that can happen, right? So it's very good. It's very useful to have an alignment scoring scheme. So this actually is kind of a pretty simple scoring scheme. So if there is a match, there can be like so. So if you are matching a sequence of, say, n, which is greater than m. So m is the sequence of your read that you are matching, and n is the sequence of your reference. So your reference is much bigger, and your read is much smaller. Now, if you want to match that read to a reference genome, it will match in multiple places. Now, all those places you cannot say that that read generated from all these places, right? So what you need to do is to find out a way to quantify this alignment procedure, to say ki this alignment has been done with this particular score, and that alignment has been done with a lesser score. So this alignment is better than that alignment, like that. So you need to have some sort of scoring system to quantify which alignment is actually the better alignment, right? So this is a pretty simple scoring scheme. If there is a match, you get a +1. If there is a mismatch, you get a -1. And if you consider a gap, you are actually penalized for it with a -2. So let's take these two stretches. So one is a 4 letter string, and another is a 3 letter string, right? So these two can be aligned in two different ways. So in the first case, you can have a gap in the middle. C and C matches, A and A matches. In the middle, A and G does not match, and there is a gap inside A, just below A. And there can be another type, another way to kind of align this sequence to the reference one is basically you align A and A, then you align G and C just after that, and there is a gap. So there are these two are possible alignments for these two particular strings, right? But which is a better alignment? That can tell you the scoring system or the scoring matrix can tell you which is actually the better alignment. So for example, in the first one, there's an A and A match, so this will be a +1. Then there is a gap, this will be -2, and there is a mismatch, which is -1 again, and there is a match. So if you add that up, that comes us to -1, and if you apply the same scoring scheme here, it comes up to -3. So this, the first alignment, the score with a -1, is better because it has a better alignment score according to your scoring scheme than the second one. So you can confidently say like the read came from this part of the genome, not that part of the genome, or this is how the read is aligned, not that, not like this, right? So the optimal alignment that you want to find, this coding system can help you do that. Now, this is a very simple scoring system that I have given as an example here, but there can be multiple very complicated scoring systems. We'll not cover those, but you can read about those. So, yeah, there are specific matrices which can give you specific scoring criteria. So, if, say, for example, you are considering a mismatch as a penalty of -1. Now, you may consider a mismatch between a purine as -1, as a sorry, a purine pyrimidine as -1, and a purine as purine pyrimidine as -2, and a purine as -1. So these kind of granularities you can like incorporate in your scoring system, which will help you get a better alignment, right? Okay. Now, the most basic way to do this alignment will be the brute force solution. You iteratively try to match that sequence in all parts of the genome or all parts of your reference and try to find the best alignment. Now, that will be a very brute force way of doing that, and it also will take exponential time. Now, if it is taking exponential time with 3 billion strings, it's actually never going to finish. In reality, if you try a brute force optimal alignment algorithm with a human genome, it will never finish. It will not finish in finite time, right? So you cannot use this kind of brute force algorithms. We'll use some more sophisticated algorithms, right? Okay, before we go into the different algorithms that are used, let's talk about BLAST. I think a lot of people have here are familiar with BLAST, and many people have used it. Uh, sorry, yeah, many people have
No need
used.
to apologize.
Yeah, okay. So let's see how BLAST works. So BLAST, how does BLAST work? You give a sequence, query sequence in the query box inside BLAST, and you click on BLAST, and it tells you like what are the most like optimal reference genomes from where this particular sequence originated. So how does BLAST work? BLAST works by comparing query sequence to a database of sequence to find regions of similarity. So this is also a very heuristic approach. So what it does is it does this in 3 or 4 steps. So in the first steps, it creates a lookup table. So it's basically, what is a lookup table? It's basically short stretches of the genome, and it also, along with the short stretches, it has an index keep from where that particular short stretch generated, right? That is what is called a lookup table. You can just query the lookup table. You can go back and check for the sequence, and when you find the sequence, you can know from where the sequence has been generated, right? So that is what is called a lookup table. uh, yeah, it first creates a lookup table or a list of words from the query sequence. This is called seeding, right? And then BLAST takes the query sequence and breaks it down into small sequence called as words. For protein sequence, each word will be a 3 amino acid codon, sorry, not a codon, the 3 amino acid long. The codon is the DNA side of the uh, protein, right? Like before it has been converted to an amino acid. That is a codon, right? This is not a codon. This is a 3 amino acid sequence long stretch of DNA, stretch of protein. And for DNA sequences, it's generally 11 nucleotides long. So what it does is it breaks this whole query into small overlapping small queries, right? So for example, GTQ, then TQI, QIT, like that. Overlapping queries, and it stores these queries in a table with indexes, as in where from it generated. Similar is the case for nucleotides, right? In the step two, what it does is it uses in the next step, it searches a database of known sequences, as the lookup table is there with it, so it has the known sequences to find any sequence that contain the same words as the query sequence. This is done to identify database sequences containing matching words. So it kind of tries to match these particular words to each of the databases that it has for different sequences for different organisms, right? Then it does that optimal alignment scoring thing. So in this case, while trying to do optimal alignment, it uses majorly like for BLAST, the the scoring system is defined from particular matrices, right? So PAM is a very widely used matrix for protein amino acid basically alignment or protein alignment. So this PAM matrix, I think you have heard of it, and it's used for protein sequence alignment, and also BLOSUM. BLOSUM is block substitution matrix. So these are actually scoring matrices, right? So what are the score? What is a scoring matrix? This is how a scoring matrix looks like. So if there is a match, there's a +1. If there's a mismatch, there's a -1, like that. So like this is actually pre-generated or like predefined by BLAST, and it actually uses that particular scoring scheme to find the optimal alignment. And finally, it extends the words in both directions. So if there is a match, it will try to find more matches in that region. So it will extend the words in both direction while counting the alignment score using the substitution matrix. And if the score will drops below a certain threshold after extending, it will uh, assume that that is a wrong alignment, that is not a good alignment, so that it will not take. But the one that is the most optimal one, it will take, and without gaps, if the resulting aligned segment is without gaps, it is called a high scoring segment pair or an HSP. And for each of these alignments, BLAST will give you an E value. It is very similar to a P value. People who are uh, like familiar with it, it's basically tells you a significance, how much significance is that particular event occurring in. So basically, if it is giving you an E value of 5.05, what it means is that it is 5% chance that the optimal alignment is not the correct one, right? So it's very similar to a p-value, but here it's called an e-value, right, for BLAST. So now, as we saw in BLAST, what BLAST does is it breaks down your query into small sequences, and then it creates a lookup table and indexes your sequence. Now this indexing concept is actually quite old for humans. So if you see random books, you turn it to the last page, there will be an index of words. Similar thing we are trying to do here. So there can be two types of index. One is the forward index. So for example, the word and where it is presented. So document 1, 3, 4, 5, 7, like that. And there can be reverse indices as well. So reverse indices is basically, or inverted index is basically, this is the document and these are the words present inside the document. So the type of indexing we are interested in is basically reverse indexing, and this is the inverted index. But we simply refer it to an index. So how are we trying to index? So this is, say for example, there is a stretch of the DNA. What are the different uh, small queries that can arise from this stretch of the DNA? That is what we are trying to say. So in this case, we are saying document 1 contains these words. In our case, what we are saying is this stretch of DNA contains these bases, and these are its substrings. So this is what we are trying to do. Right? Just give me a minute. Okay. Yeah. So this is how the indexing works. So you have a long string, and then you break it down into small substrings, as BLAST was doing similarly, with some sort of overlap. So in this case, the overlap is 1. You can have more than one overlap. You break it down, and then you sort it and keep it in some sort of data structure. In the, in in practical cases, suffix arrays, suffix trees are used, but you can assume that you can keep it in any kind of data structure. So for example, what information can you get from the data structure? Okay, this is actually a substring of the whole sequence, and this substring starts from the 4th position. So this one. So CCTAC actually is a substring of this whole string, and it starts from the 4th position. Right? And there can be like multiple so a particular substring can be present in multiple indexes, right? So for example, this CTTAC is present in this position, which is the 8th, and also the 12th position. So these kind of information you can, so what you're doing is you're breaking down the whole string into small substrings, and you're saying where this particular substring is originating from in the original string, sorry, in the original string. That is what we are trying to do using indexing. uh, like I'll not go into this. Okay, okay. Let's discuss this actually. I thought I'll skip this, but okay. Say for example, you have a long text like this. You have a long text like this. What you want to do is create this kind of a table or a data structure which can tell you what are the substrings of this particular long string and where are these particular substrings getting generated from, right? So how you do that? So the technique is you add a dollar sign at the end of the string. It's given by this $. So this is your string. You add a dollar sign at the end, which is acting as a delimiter, which is basically telling you the string ends there, right? And then after that, you create all the substrings of this possible string. This is your original string, and these are all your possible substrings, including the last one, which does not have any other character other than the delimiter that you have used, right? So these are all the possible substrings of this whole string. Now, what you are trying to do is basically build this into a data structure. So what this particular data structure is called is a suffix trie. I'll not go into very detail of it because I see a lot of people with like only biology background, so I'll not go into details of the computation stuff behind it, how we can optimize the time and space complexity. But I just want to give you an overview of how these things work, how indexing works, and how can indexing help you do the alignment, right? So this is how the trie will look like. So basically, each of these parts that you see is actually spelling out a particular substring. If this is your longest substring, the green one marked, which basically is the whole string itself. That is the longest substring, and your shortest substring is only the delimiter, like this. So this is how a suffix trie is formed, and at each position you can have the index of these particular substrings that is getting generated. So you can save it in a data structure which is simple and yet elegant. So in this data structure, you can find all the strings that are all the substrings that are part of the original string and at what position this particular substring is present. So yeah, as I was saying, the green is the longest substring and the blue is the shortest one. Right. Now, if you do not add this dollar sign, what will happen is you'll have a lot of repeats because a substring, say for example, let me see if I can find any repeats. Ha, let's see. Consider the last four, right? GCGG, right? And consider a substring here as well, GCGG. Now these are two same substrings, but they are originating from different parts of the string, right? If you do not add this dollar symbol at the end, or if you do not add a delimiter at the end, what you'll end up is something like this, which is quite different from this, right? Here you can see all the substrings very clearly, like distinct substrings very clearly, which is not the case in this kind of thing, right? So if you do not add the dollar sign, it's actually quite messed up, and the same substrings that are getting generated from the original string will have the same kind of indices, and you will not be able to resolve this. So that is why using that delimiter is important. Now each will be labeled with a single character, and the nodes have no label in our representation. But if you think of this node is actually the stretch of the sequence coming from the root to that particular node. So this is actually BAA. So this node actually represents BAA. We do not write it like this inside the node, but this is what it actually represents. Right. Now, this data structure is called a suffix trie, and it is an ideal data structure to quickly check if a pattern is present in a text or not. Now, coming to our alignment problem, what was our alignment problem? We have read. We want to check if that read is present in the whole genome. The same concept is here. We have a pattern, we have a text pattern, which is a substring of the original text. You want to see if that pattern is present somewhere in the text or not. So the problem is same actually. So uh, this is a very good way to do it. Now, if you want to check, say for example, a particular string, say for example, BAA is present within the text ABAABA. So this is your text and this is your substring that you are trying to find in your text. So in our context, this is our reference genome and this is our read, which we are trying to align it to the reference genome, right? So this is represented in this suffix trie. What we do is we start at the root and take an edge labeled with B. So we start a root and we see all these paths. So there are three possible paths to move from the root to the next node. One is through B, one is through A, and one is just the dollar sign. So since our pattern spells BAA, we take this route, right? We take BA, right? Now we start with B. Now, next we proceed to an edge labeled A. So from here, you only have one option, that is A. So you can proceed over here. If there was not an A here, then that might indicate that that substring is actually not present in your suffix tree, right? So, but you have it here. So you move to the next node. At this point, there are two outgoing edges, which is A and dollar. Since the last character in A is A, we can take the edge labeled as A. Because the entire substring BAA can be spelled out as a path from the root, we conclude that BAA is indeed a substring of ABAAB. So this was your original string, original text, which you converted into a suffix trie. Then after converting it into a trie, you start from the root and you try to trace a particular sequence or the pattern, in this case, which is BAA. And if you can trace that particular pattern from the root to some particular node, then you can conclude that actually that is a substring of the original text. So in our case, this whole trie will be made up of your reference genome, and when you're trying to find a particular read inside the reference genome, what you'll do is you start from the root, and one by one in the sequence, you'll try to go from one node to another. And if you can go from the root to a particular node inside the suffix trie and cover the whole read that you are trying to find, then you can conclude that that read is present in your own genome. And that is and also these will also be marked with the indexes of the positions from this particular read generated. So you can also claim that from this position, this particular read generated, and this particular read is present in our genome because we can travel from the root to a particular node inside a suffix trie for this particular sequence. Right? Okay, so that is actually the whole idea behind alignment. So in all the alignment, there are various alignment softwares. Tomorrow we'll use the BWAMM, but yeah, there are all sorts of alignment softwares. But the basic idea is similar. That you have some sort of a data structure. In this case, it was suffix trie. You can have suffix arrays as well. If you want to convert a trie to a tree, it's basically you collapse all the non-branching nodes into one. So it's basically a more compact representation. So basically, these two nodes have been collapsed into BA like this. These three nodes have been collapsed into ABA dollar like this. So it's a more compact representation, but the data structure is very similar to a trie. Uh, as I was in comparison with a trie to a tree, the only difference in finding a particular substring in a tree will be that you might have like half match inside a particular edge. So, for example, you are trying to find BAA inside the genome. From the root, you move to this node because this starts with B and then follows with A. So you move to this node, and in the next one you cannot fully move to the next node because there is an ABA dollar. You just want a but so that is what it is, actually half matching. So this thing you have to consider when you are using a tree in comparison with a trie. But using a tree is a more compact data structure, and it will save you more space. So tree is used more often than a trie, right? Okay, I'll not go into this. This is a little bit more in detail, but yeah, suffix arrays are also like similar kinds of concept of suffix tree or tries. Instead, we do not have a like a root to node data structure. It's just an array where you can store. So this is your original string, and you can break it down into small substrings, and you can store the pointer variables here at what index is that particular substring generated from. So this is a much more compact data structure compared to a tree or a trie. Say, for example, if you take a human genome, a suffix array will occupy around 12 GB. And if you use a suffix tree, not even a trie, if you use a suffix tree, it will be 47 GB. It's like 4 times. So it's actually pretty useful to use suffix arrays in place of suffix trees or tries. Right. Okay. So Barros-Wheeler transform today, I'll not go into it because tomorrow we'll do BWMEM, and then we'll discuss this concept of Barros-Wheeler transformation. But yeah, we are almost towards the end of the session. But before I leave, I just want to cover couple of topics. So basically, the previous topics that I was talking about is basically the sequence alignment problem and how you can what are the different types of algorithms that we use for finding these particular short stretches of DNA in the whole reference genome. Now, after this alignment has been done using particular softwares, what you generate is a SAM file, which is sequence alignment format. So it's basically it's a very similar data file to a BAM file. I hope you have heard of a BAM file. It's actually SAM and BAM are actually very same. It's actually pretty same. Just in case of BAM, a binary is used the data is stored in binary form, so it actually compresses the data size by a lot. So it's your data will be generated as a SAM format, but you can compress it further by converting it to a BAM format, right? So yeah, most SAM format data is output from aligners that read FASTQ files and assign sequences to a position with respect to a drone reference node by the algorithms that we just discussed. So yeah, for the alignment algorithms like alignment software, sorry, like BWMEM, it'll take the FASTQ file as an input, and as an output it'll give you a SAM format file. So just I want to give you what a SAM format is and what information you can see here. So yeah, as I was saying, yeah, SAM and BAM are the same, and just it's in a binary form. BAM is a binary form of SAM. It's same. Okay, so what you have is in the original data file, you'll have a lot of information. So let me just first show you what is how it looks like. So this is your header line in headers, and this is your alignment portion of the file for the SAM file. So a lot of information can be incorporated here, but the most important is the sequence here, is the seek which is given here, and after that this is the quality score. If you see, from it starts from this 0 and ends at this greater than sign. So this is also ASCII coded. So this is actually is representing the quality score for each base using ASCII characters. So if you converted it into numbers, it'll actually be like 20, 30, 40, something like that, which are actually Fred scales quality scores, as we discussed. Yeah. So yeah, this is how the header looks like. So what information can you get from the header? A lot of stuff actually. Uh, uh, Sorry. Yeah. It contains the version of the SAMWAM file you're using. It contains supplemental information for alignment records, like information about reference sequences. Information may point. It also may point to this supplementary information, identifying which ones of the specific alignment it is associated with, and it can have other information as well, like it can have our names, cigars, and those stuff. Those stuff it can have. So, but the main important thing is in this part, the alignment section, and this is the sequence that has been aligned, and this is the quality score it has been aligned to. And this is MPOS is the position of the alignment. So this is giving you actually the position at which a position of the reference genome at which this particular lead has gone and aligned. Right? Yeah, I think that's all from my side for this class. If you have any questions or doubts, I think now is a time to ask. Just before you ask, let me just go back to this particular slide. Uh, so what have we covered today? We covered different types of sequencing technologies. Then we covered how you can generate quality scores for each of the sequencing base that has been sequenced. After that, how we do preliminary data quality checks like adapter trimming and removal of low quality bases. Then we talked about the sequence alignment algorithms and how these work. And after that, like what is the output of the sequence alignment, which is basically the SAM and the BAM format. After this, we'll move into variant calling and how do we do variant calling, what are the algorithms, what are the softwares used, and we'll have a small hands-on session tomorrow as well. So yeah, and if you have any questions, please do ask. Okay, Python packages. There are Python packages. I'm not I'll not say they are not, but Python is actually not a very good way to handle this this huge sets of data. So we generally do not use Python to actually kind of analyze uh, do this kind of processes. So the data quality check, as I said, or the sequence alignment that I said. Uh, yeah, the no R also like the I'll tell you the issue with Python and R. We use Python and R, but if you see the softwares actually that we use for alignment, say for example BWMEM or GATK modules that we use, those are actually written in C++ or Java, stuff like that, because uh, we have to optimize space and time very carefully because the amount of data is huge and the time taken to do this stuff is also huge. So generally, these algorithms are not written in Python. These are generally written in C++ because it's a faster language. But in Python and R, also you can do this stuff. So there are packages. Uh, I let me see if I can find them. I do not particularly use like we use what to say Python and R, but for that is for a downstream data analysis, maybe after alignment, after we've called the variance. Now, if you want to see particular things about the variance, or if you want to do particular analysis, then we use R and Python. We generally do not use R and Python in these steps. So there are bioconductor packages in R which you can look through. So I am not pretty confident about what exact package you can use, but I think in bioconductor you can find some packages. From what I remember is G ranges, I ranges, those stuff. I don't actually remember R and Python packages, but yeah, you can what you can use is Galaxy is a very good tool you can use. So in Galaxy, you also have example data sets, and you'll have access to a lot of algorithms, a lot of softwares that are uh, uh, inside the Galaxy software also. So you can use those to kind of practice on your own. Uh, yeah, BioPython and Bioconductor. These are the two major uh, things. Yeah, yeah.
Thank you so much, sir, for the clarification.
Okay. Yeah, so what I'll do is I'll upload this actually I've recorded this whole lecture, and I'll upload this on YouTube, and I'll see if I can and most probably in the next class I can give you the link to this. I mean, tomorrow I can give you the link to this video, and you can take a look later if you want. But yeah, is there any general questions about the things that we discussed today, particularly the algorithms, the different methods that we use? And please do give feedback on on kind of the technicalities that are involved. So for example, I did not go too deep into the alignment stuff because if you want to, from a computational perspective, there is a lot to be discussed in terms of space and time complexity and how we can optimize those. So I did not go into that because I saw the maximum of the people are from biotech or bioinformatics or molecular biology background. So I did not go into that. I hope that's okay. But yeah, like do give me feedback if I like should more focus on the biology part or the computation part, or is this okay going on for tomorrow? And yeah, if you have any questions, please do ask. Actually, if you guys, I don't know if you are familiar with Langmuir. So uh, he's actually a professor of kind of you can say bioinformatics or something in MIT. So he takes actually a pretty good. It actually takes up meaning like kind of all these classes in MIT, and those are actually recorded in YouTube and present in MIT OCW. You can go and check out his videos. Ben Lengmiat. I'll just send the name if you want. Uh, no, it's actually from a molecular aspect because uh, it's not from a software and data analysis aspect because we have enough amount of like kind of computational technologies to kind of have a long stretch of DNA sequence at a while. It's the chemical part that is actually not feasible to do so because there are multiple factors involved, but the the main thing is uh, like it's very hard to kind of this sequencing by synthesis process. Why ONT, as I said, is giving you such big reads is because it is not using sequencing by synthesis process, but still it cannot give you kind of like in the level of million bases or something. It still is stuck at 10,000 because like it's very hard to kind of do this thing for a whole stretch. Sequencing by synthesis, it's very hard to have long stretches of DNA. That's why in most cases we see short stretches because after a certain point of time, same as the issue with Sanger, it's very hard to differentiate between. So it is easy to differentiate between a base at a 10th and an 11th position, but it is not easy chemically, I'm saying, but it is not easy to chemically distinguish a base from uh, a million to a million plus one position. So chemically, it's hindering, not from the data analysis perspective. So yeah, it's actually the other way around. So this is actually website, so you can take a look at it. And someone asked something else, I think. Okay, no. Okay, then if there are no further questions, I'll close this session and I'll see you tomorrow, same time. Thank you. Thank you all for joining.